nematode ecology in natural systems, and got her PhD in biological sciences in Lancaster University in the UK in 2007. Then she returned to Portugal and she continued to do research on nematode ecology, focusing and applying the ecological theories developed that she developed in natural ecosystems, and she started applying them to agroecosystems where plant-parasitic nematodes cause major losses. So in 2015, Sofia joined CBMA, and her research now follows two main lines, converging in the topic of agroecology. So the ecology and rhizosphere interactions of plant-parasitic nematodes with other organisms, and second, the use of soil nematode communities as bioindicators of key ecosystem services. So by the end of this communication, all of you will be aware of how fantastic animals nematodes are. So Sofia, please. And thank you very much for accepting to give this seminar.
Thank you, Celia, for the introduction. And also thank you for inviting me to do this talk. I'd like to talk about my work. I'm going to give you an overview of what we've been doing in CBMA in the nematology group. Not many people have heard about nematodes, and those who have may have heard about nematodes very superficially when they're studying zoology and looking at nematodes as another step in the evolution and phylogeny of organisms. So I'd like to give you an overview of what nematodes are and what nematodes do and how that affects us. And by the end of this talk, as Celia was saying, I'm hoping that you'll get a glimpse of how each of us is affected by nematodes and their roles in ecosystems, both directly and indirectly through their different actions. I'm also hoping that by the end of the talk, you'll see that no matter what research line you're working on or hopefully going to work on in the future, you can use nematodes as organisms of interest because they're involved in so many different processes, and there's so much we can learn from them, both from their ecology and also on their biochemistry and metabolism. So to give you an overview, I'm going to talk about nematodes in general. You'll see that some are beneficial, so we have the good, and we have the bad ones that affect us negatively, and supposedly the ugly. So the most that we see here and also there is the same one. Here you can see that it's been photographed using scanning electron microscopy, so it looks opaque. But nematodes are transparent. As you can see here, this one is munching away at a lawn of bacteria, of E. coli. Caenorhabditis elegans is not only the most well-known nematode, it's possibly one of the most well-known organisms. It's a laboratory organism. For some time, people have been culturing these nematodes and using them as model organisms to learn more about other animals, animals in general, humans even. And for a long time, all these lab cultures were established and used for different biological purposes. And at some point, somebody asked, do we know about any Caenorhabditis elegans in their natural habitat? So aside from petri dishes in lawns of E. coli, has anyone ever seen this? And it wasn't until a few years ago that by looking for them, people actually started to find them in their natural environment, which is fermenting fruit, like rotting food in like grapes and fruits and things like that. These nematodes are very useful because they can be grown axenically, so in cultures in petri dishes. They have very short life cycles. So if you're doing genetic studies, it's very easy to follow up what's happening and then extracting them, or just simply staining different metabolites that you're interested in, and you can, because they are transparent, you can follow them along the organism. They have a limited cell number, and actually from the egg until the full organism, you can map, you know what each cell is going to turn into through their embryonic development. They're very useful neurobiology models because they're connectome. Each neuron, we know each function of each neuron, of where it is and what it does. They've been used for aging and apoptosis studies. They have, like I said, short life cycles, but they can enter dour stages, resistant forms that prolong their life virtually, not forever, obviously, but for a long time. And that got the Nobel Prize for Medicine in 2002 to researchers, possibly the only researchers working with nematodes. They got their Nobel Prize. It was the animal where mitosis and meiosis processes were first described. It was the first animal to have its genome sequenced back in 1998 by the Caenorhabditis Sequencing Consortium. And it was the first nematode in space that was sent there, especially and precisely because of the dour stage, this resistant stage, to see how then these nematodes came back to life after the resistant stage was over, and how muscle development took place, and how muscles were affected by these no gravity systems in space. So Caenorhabditis elegans is very interesting, and it's a very interesting model for work in research, but it's not the only nematode, obviously. And once people started looking for them, and this is something that I was involved in as part of a large consortium of over 70 international institutions, we started looking at nematodes in soil. Nematodes are very simple animals. They're microscopic animals, most of them, and they colonize any sort of environment. I'm only going to talk to you about soil nematodes in this seminar, but please be aware that there are nematodes that affect us even more directly, like parasites, which many people will have heard of, human parasites, animal parasites. Then there's nematodes in water as well, in the oceans, in the riverbeds, that I'm not going to talk to you about. But if we only look at topsoil, the first superficial layer of soil down to 20 centimeters, you have this amazing number of nematodes in the planet. So they're very abundant, and you can, if you look at their distribution, which we got from modeling of contributions of nematode numbers throughout the globe, you will see that in the tropics is not actually where you have the most abundant nematodes. They're not, the population density of nematodes is actually higher in tundra and taiga biomes. So they're further north in these very cold regions. And this is surprising because for most animals, and that we know of, usually that we look at above ground, they're more abundant in the tropics. But if nematodes have these sheer numbers, they're the most abundant animal in the planet, and they're more abundant in the Arctic regions, in these cold forests, then this totally reshapes our knowledge of zoology and patterns in zoology and animal distribution. And this is very important because even though they're microscopic animals, because they're present in such large numbers, they have an amazing biomass as well. So we're talking about something like 300 gigatons of nematodes in the planet in the first soil layer, in the top soil in the planet alone. If they're more abundant here in these cold regions, then they're going to be more active in climate change scenarios that we think of, where this becomes a little warmer. And because nematodes, their life cycle is very dependent on temperature, all these nematodes that are in these regions and that are mostly dormant most of the time. They're very slowly moving. They're very slowly decomposing all this organic matter that keeps accumulating because it's too cold to have enzymes working to break it down. But if the planet warms up even 0.5, 1 degree, at worst case scenarios, these nematodes are going to be so active. They're going to quickly metabolize all this organic matter. They're going to bring in a lot more CO2 as they're doing it. So the consequences of this is a snowball effect. It's going to be tremendous. So I'm telling you about all these nematodes, the most abundant animal in the planet, but they're not all the same. If you start looking at that, what these nematodes are and what they're doing, you'll know that it's a large stroke. You'll see that nematodes can be divided by their trophic group, by what they feed on, their feeding group. So many nematodes are feeding on bacteria, like C. elegans, fungivores, also nematodes feeding on plants, feeding on mostly anything, omnivores, and nematode predators that feed on other nematodes and other soil organisms here. So if we start thinking about what they're doing then in these different trophic groups, you can start to have an idea of what they're doing in these ecosystems. Looking at soil, soil is not independent from the above ground ecosystem. Obviously, plants are the obvious link between the two, below ground and above ground. It's all the same ecosystem. They're all interlinked. So if you look at nematodes in soil, in the soil food web, and no matter where you start, if you start at the bottom of the food web in producers, plants, you can see that some nematodes feed on plants, the herbivores, yeah? And then they get fed on by other levels. And if then all these plants are producing primary production, they're producing nutrients that these nematodes get, but herbivory, what this biomass that plants keep producing, only about 10% of that goes into herbivory. Most of it is not eaten by anyone, yeah, unless, at least when it's fresh. This freshly produced biomass is not eaten by herbivores. 90% of it just returns to soil as dead material. So actually, most of the energy in these soil food webs comes from organic matter. 90% of it is organic matter. So if you start looking at organic matter and how it is decomposed by fungi and by bacteria, and if you follow those food chains, by fungi, you get nematodes feeding on fungi. From bacteria, you get nematodes feeding on bacteria. And then you have nematode predators that are feeding on other organisms. And this is just to say that any food chain that you start following in the food web, you'll come into contact with nematodes. Nematodes are involved in various processes through their feeding. So they're not only present anywhere in all biomes, being the most abundant animals, they have very different functions, very different roles. They participate in all these soil processes. And once we get an image of what nematodes are doing, even if you only look at nematodes in these soil food webs, you will start to learn what processes are taking place. Because as nematodes are feeding, you know that, okay, if you have lots of herbivores, that means you have a lot of activity from plants. If you look at nematode fungivores, you'll know that decomposition is important, and especially fungal-based decomposition. So you can start to tell that these nematodes are, if you have a fingerprint of these nematodes or a sort of a snapshot of nematodes in the system, you learn a lot from them about what's going on in that system. And so nematodes have been proposed and used for a large number of years now, since the 1990s, as bioindicators. What do they bioindicate? They bioindicate all these soil processes that are taking place. But if you want to use them as bioindicators, you shouldn't really stop at their feeding group. The more resolution, taxonomic resolution you get from them, the more you can attribute them into functional groups, into functional guilds. And nematode guilds involved in different functions depend on not only their feeding group, their trophic group, but on their overall life strategy of being mostly persister or mostly colonizer. This has to do with their metabolism. Do they quickly multiply like C. elegans? C. elegans, if you feed it bacteria, it will explode its population numbers. Yeah, it will reach very large levels quickly. But if then you starve it, they're all going to die. Yeah, some of them will enter resistant forms, but that's it. That's their life strategy. They're colonizers. They rapidly use up what's there in the medium. And then you have persisters that have very long life cycles, and they persist. These nematodes, and you can imagine microscopic organisms, transparent organisms moving around in soil, lasting 2 years. Some nematodes have a lifespan of 2 years, and they're the persisters. They grow slowly, they feed slowly, they have a few offspring each year. They don't really invest in explosive population dynamics. So if you reach the functional guilds, you get a wealth of information about these nematodes, and then you condense that into indices, ecological indices that I'll show you in a moment. But what's important is that because so many people have been using nematodes as bioindicators in various different ecosystems subject to different impacts, you know what their response will be from what's there in the literature, what's documented in the literature. So you know that, for instance, if you look at soil as a habitat, if you then plow it, you know what's going to happen to nematodes. And if you don't know what happened in the soil, if you look at nematodes and how they're responding, you know what kind of impact you had. And this, then you have a lot of computational, you need a lot of computational tools to help you condense all this information of a nematode community and all the functional guilds and their strategies and their numbers into a data point in a graph. In this graph, like the faunal profile, just you get all these indices calculated and you end up with a data point. And if your data point ends up in this quadrant, you'll know that your community is structured. It has complex food webs, long food webs, long food chains in complex food webs that self-regulate. Yeah, so all this diversity and complexity in food webs is a regulatory mechanism. And if you end up on this side, it's very basal. It's very, the nematode community in the food web has been stripped down to its most basal point, most impacted point. And here on the y-axis, you can go from completely nutrient depleted, so no nutrients available in this soil. So very, these soil communities, these soil food webs are starving. Or you can go to enriched conditions in which you have so many nutrients available in the soil food web. So depending on where your data point ends up in these quadrants, from one nematode community, from one soil sample, you can tell a lot of what's going on in that system. Okay, you can tell if it's disturbed. So it's got a lot of nutrients, so you've got a lot of enrichment, but basal communities, very simple food webs. That means there's been a lot of impact and there's lots of nutrients, but they're not really contributing to complexity. Yeah. So this is not a very good point to be, because you have lots of nutrients, but they're not really being integrated into regulation, into complexity of the soil food web. Whereas here you see there's not many nutrients available, but there's been a lot of investment in complexity, in structure of the food web. And because this not having lots of nutrients, it doesn't really mean that there aren't many nutrients. It just means that they're quickly taken by what's there. Yeah, all these nematodes, all these organisms are quickly taking up all these nutrients, so they're not freely available. And like I said, Nematodes, the different bacterial feeding groups or fungal feeding predators, herbivores, omnivores, they have different metabolic rates. They have different metabolism depending on whether they're colonizers or persisters. So if you add a level of complexity here and add what their metabolism is like, you can come up with these amoeba plots, these radar plots, in which you know from your nematode communities, you know where the nutrients are in the soil food web. So if they're here, if they're from 0 to 5 in any direction, but if they're here, all these nutrients are being put into herbivory. All these nematode herbivores are taking up all that's being produced by the plant. And this reveals pest pressure, nematodes as pests, yeah? So herbivory in this system can be quite high. If it is so, this is a disservice, yeah? But you can have nematodes, bacterial and fungal feeding nematodes on this side of the plot. If you have a lot of activity from bacterivores and fungivores, that's where the nutrients are going to be. That means your mineralisation service is high. So that means all these nutrients that are coming in are going into decomposition. Decomposition of organic matter is needed, of course, because plants cannot absorb organic matter. Plants only get their mineral nutrients. And to get mineral nutrients from organic matter, you need all these organisms to decompose it, yeah, to mineralize it. So it's a mineralization, it's an ecosystem service. And then you have another key ecosystem service of regulation. So if you have a lot of omnivores and predators and they're very active in the food web, that means they have enough food, there is sufficient food, and there's sufficient stability in the ecosystem to develop very long food chains, to develop food chains that accommodate predators and omnivores taking up this energy. So it means that the food web is complex, and it means that it's regulating. So you've got a regulatory service of the ecosystem based on nematode ecology, on the nematode, on the soil food web. So this is all very theoretical, and I'm going to give you some practical examples of what we've been doing using nematodes as bioindicators of ecosystem services. And before I showed you, sorry, just moving all the faces to a different side of the slide here. I've shown you before this contribution that we added to the international consortium looking at nematodes in the globe. And we were only looking at feeding groups. Now we have gone into functional guilds. And we've collected all this data from different ecosystems in Portugal as well, that we've compiled to send to our colleagues that are now gathering all this data, not feeding groups, but functional guilds, and trying to get a lot more information on ecosystem services across the globe. So that's a paper that's in preparation now by Stefan Guyson. So we went back to the real world out there, and we took samples from oak forests, from sand dunes, from eucalyptus forests that are very fragmented, so not only eucalyptus but also pinus as well. And then we took samples from agricultural systems that are more impacted, and we came up, again, if you remember the graph here, what you have is a structured ecosystem, a structured food web, and lots of nutrients available. And if you look at all these points, and I'm not sure you can read the legend there, but looking at different ecosystems, you have the forests here and the sand dunes, they tend to be less nutrient enriched. And that's actually a good thing if you have structure, because that means that all these nutrients are being used very rapidly. And here you have agricultural systems that are more enriched because people fertilize them, okay? But you have two very different ecosystems here. You have extensive agriculture, and this is open field crops that are not very impacted with pesticides. And you have greenhouse agriculture. That's, you know, greenhouses are like small scale scenarios of global change, because they're warmer, they're more saline environments. They you know, they have a lot of impacts. And you can see that here, yeah, you have the point in the middle, and then you have what the metabolic footprint of this is. And you can see that this is very square shaped, and this is spindle shaped. And this tells us that all these nematodes can use nutrients. They're very specialized in nutrient, they're colonizers, they take up nutrients rapidly, but they're not very structured. Yeah, okay, structure is there, we're on this side of the plot, but the community itself does not allow for a lot of structural change, only nutrient uptake. And if you look at then where these nutrients are, looking at the metabolic amoeba, of course, different systems are going to have different nutrients going into different services. So you have the decomposition service here, a regulatory service there, and the herbivory disservice is, of course, much higher in agriculture and much higher in greenhouse agriculture. So you see a lot of pest pressure in these ecosystems. And this is the disservice that you don't want, basically. Looking at then some work that we did, and we had an MSc student, João Souza, looking at this. We started looking at, okay, a sustainable agricultural ecosystem, and that is in the Barroso region, in Montalegre. This actually qualifies, it's the only agricultural system that we have in Portugal that's been certified by the Food and Agriculture Organization of the United Nations, as agricultural heritage. It's a globally important agricultural heritage system here in Portugal. Why is it so important? This, for centuries, has been managed sustainably. People have a lot of dependence there on cattle, and you can see here the Barrosin cow. They're meat cows. They're cattle used for meat, and they graze in these fields, so that you have pasture here. Now, these animals are not there all year, and they go to their stables, and of course they poo everywhere. They eat a lot and they poo everywhere. And what people for centuries have been doing is going to the nearby natural ecosystems and collecting all these bushes, all this brush, and breaking it to make animal beds. And these animal beds are where these cattle then stay, and they poo in it. And then all this material, this very rich organic matter, is left to decompose a little bit, and then it's spread on the fields where you have your agriculture and you have your nutrients. You need your nutrients, and they're coming from this manure, this composted animal bedding. So it's circular, yes. So you have nematodes, cattle grazing, and you have all this organic matter coming from natural systems for the animal bedding, and then that, once it's enriched with all the animal ingredients, all this nutrient, this manure, then gets spread on the fields, and that's where agriculture takes place. And João started looking at this. He took a number of samples, and if you look again at the graph, you see that in natural ecosystems, they're all very structured. Remember, this is signaled as globally important, sustainable system. You can see they're all structured, they're all complex food webs, so not a lot of impact, but you have a lot more nutrients in your agriculture and pasture, and these nutrients are coming from cattle and combined then with the all this plant material coming from the natural ecosystems. So once you look again at the metabolic footprint, you can see again that in agriculture, Most of the nutrients are going to herbivores. These are nematode herbivores. These are pests in agriculture. So we're interested in herbivores, plant-parasitic nematodes. Nematode herbivores are not just any random herbivore that goes in and chews a bit of a plant and goes away and takes another bit of plant somewhere else. They develop close associations with a plant, with their host plants. They're all plant parasites. They're obligatory parasites of plants. And for centuries, they've caused damage to agriculture. But also for centuries, farmers didn't know they were there. Remember, these are very small microscopic organisms in soil, and they don't cause specific above ground symptoms. So farmers would notice that their plants were growing poorly, they were wilting, they had chlorosis, so they were yellowish. Yeah. If you water all these plants and fertilize them, they wouldn't respond. So what people didn't know, of course, above ground, you can, if you have a huge, very high nematode infestation, you get patchy areas where you have a high density of nematodes, and they can really cause major losses in fields. But above ground, you don't know they're there. And it's when you start looking at these roots and you start uprooting plants that, depending on what nematodes you have there, you can have symptoms specific to nematodes. And this is galling of roots. So roots shouldn't look like this. You should all know what roots look like. And all this galling, these knots, are caused by a nematode. And you can see smaller ones there. So, okay. Nematodes are perceived as plant pests in agriculture. They cause huge losses worldwide, and they're in roots. So you need to know, most of them at least are in roots. So you need to know a bit more. If you want to know a bit more about them, you need to start digging up these plants and looking at the roots, and use a good microscope as well. You have different nematodes. They're not all the same. These are all plant parasitic nematodes. Some go inside the roots. These stay there. They select a feeding site and they become sedentary. So they go fat. As you can see, they're not worm-like anymore. They become round-shaped. Same here. And some nematodes that go in the roots, they keep active, so they don't get fat, and they keep worm-like. And all these nematodes are feeding externally. So you have different groups of nematodes. And somebody back in 2013 started collecting all this data from everywhere in the world, and it came up with this top 10 of plant parasitic nematodes affecting agriculture worldwide. I'm not going to tell you about all of them. We have been working mainly with the top 3, and especially with the top 1, Meloido. Meloido is this one. It's the root knot causing nematode, so causing these galls in roots that we see here. But because we're interested in nematode ecology, we end up looking at whatever nematodes appear in these ecosystems. Root knot nematodes, oops, I'm moving forward a bit. Root knot nematodes are the most damaging plant parasitic nematode. They're present everywhere. They're polyphagous, and as a whole, as a group in the whole genus, they parasitize virtually all plants. All higher plants can be parasitized by root knot nematodes. And this makes them especially damaging for agriculture, especially damaging for horticultural crops. They're widely distributed, like I said. Their life cycle is temperature dependent, such as the life cycle of all other nematodes. And they can reach high population levels, highly damaging levels, especially in greenhouses where it's all very warm and the host is there. I said they're polyphagous, but tomato is their universal host. And we have a lot of work on this interaction of root knot nematodes and tomato that I'm going to tell you about. So, but like I said, keep in mind there's many different nematodes, many different plant parasites as well that can have damaging or negative effects on your ecosystem. I'm going to quickly tell you about, moving on to now, farming systems, cropping systems. I'm going to briefly tell you about this project that's coordinated by Teresa Almeida in our group, and they're looking at, we're looking at potato cropping systems in Portugal. There's many areas, main regions of potato production, and we focused on region 1, 2, and 4. We have one sample from region 5, but I'm going to tell you a little bit more about these regions. So again, you know about the plot. You know that this is nutrient enriched as in any agricultural system, but you see that it's not structured anymore. So once you start looking at horticultural crops, these annual crops, then you lose, there's so many impacts, so much human disturbance in these ecosystems that they've lost their capacity to regulate. So they have very simple food webs as well. So they're on this side of the plot. Okay. And if you look at the metabolic footprints, it's mostly bacterivore and fungivore. So it's mostly going into nutrient enrichment of the ecosystems, into decomposition and explosive dynamics of colonizers. And also, yes, herbivores are present. And we know all about herbivores. In these ecosystems, we found numerous genera of nematodes, and they're all these nematodes listed here are plant parasites, but they're not all distributed equally. And what we have been doing lately is trying to understand what their assemblages are. Is there a particular assemblage to potato parasitic nematodes? Is there a group of genera that usually assemble in potato rhizosphere? And we're still analyzing this data. We did come up with different groupings of what nematodes are there and how they're associating. And we've been bringing together an edapho-climatic categorization, so soil and climate categorization of these ecosystems. And we're finding that these assemblages, these plant parasitic nematodes are very much responding to geology, climate, and all these different variables. And then we also started looking some time ago now at tomato cropping in greenhouses in Portugal. So it's not open field like potato anymore. We're talking about greenhouses. And again, you know, nutrient enriched, no regulatory role of the soil food web. So this means conditions are there to quickly, if there's herbivores, they're not going to be controlled. The food web is not doing any regulation or very poor regulation of the system itself. We looked at different kinds of farms, differently managed farms, organic farms, and then conventional farms. Conventional farms here we divided into large farms that are very professional, managed. They have these very large greenhouses over 1 hectare, sometimes 10 hectares, so 10 football fields of greenhouses. And then we have smallholder farms that are family owned. They sell their produce. It's not for self-use, they sell it, but they're selling it to restaurants in local markets. And they're not, as large farms have a lot of pressure by the market itself to certify everything and to use all pesticides and fertilizers very sparingly. In smallholder farms, they don't get that regulation from the market itself. And once you start looking at different nematodes, and you can see the genera here, like Trichodorids, Heterodora, Pratylincus. So these are all genera names of different nematodes. And looking at what people do, how they're managed. Here in this part of the plot, you have a lot more use of chemical pesticides, of which metam sodium is the most toxic one. And They're getting all these pesticides, and many of the nematicides are actually being withdrawn. They're being banned because they're too toxic. And here on this side of the plot, then you have more biological pesticides like fermentates and all sorts of plant extracts that people apply. And then you have crop rotation, especially with Asteraceae plants that seem to be doing something to the plant-parasitic nematode communities. If you look only at organic farms and large farms alone, you can tell that they're differently managed and that they end up with different plant-parasitic nematode communities. But if you look at smallholder farms, possibly because they're not so regulated, they end up applying all sorts of different management practices, and they go a little bit everywhere. Yeah. So we could go into a discussion of how this food is being produced that we're eating, but for ecological purposes here, we're just wanting to say, okay, plant-parasitic nematode densities and densities of different genera of different plant-parasitic nematodes are related to how you manage the agroecosystem, your ecosystem in agriculture. And that brings us to agroecology that we've been investing a lot in lately. Agroecology is a new paradigm of doing agriculture in which you consider what we usually see as the agricultural system is just this bit, producing food and feed for animals, food for us, and fiber and fuel. And usually, or traditionally, this was what people looked at as an agricultural system. In agroecological terms, you look at the whole mosaic of all this rural mosaic of natural ecosystem, the productive part of the ecosystem, and the human population as one. Okay, this is this ecosystem here, this productive traditional agricultural ecosystem is taking in all this biodiversity coming in from natural systems, infrastructure coming from here, the culture of these people and how they're managing the land into this productive bit of the ecosystem. And so the land here is not spared from the rest of the natural ecosystem. It's being shared, and animals and plants and spontaneous, the native biodiversity doesn't know any boundaries. It doesn't know that this is a different system to that one. Yeah. So they move from side to side. They move from one bit of the ecosystem to the next. And if we can explore this biodiversity, instead of trying to save the natural system by going to do this agriculture here, and I'm going to save all the rest of the ecosystem around me. No, you're going to try to exploit this biodiversity coming from natural ecosystems to try to perform the ecosystem services that you need in this bit, so that you can then use it to your advantage. So, can you use biodiversity? How can you use biodiversity in your favor by managing your agroecosystem in such a way that you can exploit it? And we're looking at a very specific question here. Can biodiversity be exploited to control these plant-parasitic nematodes? You saw earlier that in agricultural systems, in this productive part of the agricultural system, is where you have plant-parasitic nematodes reaching high populations and getting all the nutrients from herbivory. But, it's not all about agroecosystems alone. They're involved in a lot of different natural to semi-natural ecosystems, and they're bringing about all this biodiversity. This is a slide coming from an international project that we did back in 2011. There are many different control mechanisms of plant-parasitic nematodes coming from biodiversity. Plant-parasitic nematodes can be controlled by the plants themselves, by their antagonists, like fungi and bacteria that eat nematodes, and they can be controlled by each other's in horizontal control. And I'm going to skip through this slide. I'm just going to show you this sort of ecosystem, which is not usually perceived to be an ecosystem, probably by most of you. But once you start collecting samples, and you can tell that this turf grass forms a mat of roots there, and then below that substrate is just sand. And if you start extracting nematodes, you get a lot of different plant-parasitic nematodes, and their distribution in the field and in these upper bits of where the roots are and the deeper layers, they're all distributed differently. So we're thinking they're all eating turf grass, but there's all this diversity, and they're in different places, literally in different niches here. So they may be avoiding horizontal control, avoiding competition through resource partitioning. They're eating different parts of the plant, so to speak. You can see a bit of root here. It's all damaged by nematodes with galls and these very stunted roots. So they're rooting poorly. These plants can be scraped off easily. Once you start looking at this more closely, we found root knot nematodes belonging to a species that was never reported in Portugal before. They have spores, bacterial spores here attached to the cuticle. And this is a bacteria Pasteria, a bacterium that parasitizes these nematodes. And then there's another bacteria here that we think is a bacterium that is never been reported to affect root knot nematodes. Only in C. elegans has this been found, but you can see that it's aggregating nematodes by their tails before consuming them. So top-down control by microbial enemies is something that in agricultural systems has been explored for many years. And there's this fungus, Peconia chlamydosporia, that has actually been used as a nematode biocontrol agent. Peconia is a saprophytic fungus. It can feed on just organic matter, and we have characterized this. It actually can use lots of different carbon sources. These are all different carbon sources that the fungus can feed on, but it can switch to a parasitic phase and eat parasitized nematode eggs. These are small nematode eggs that you can see here being consumed by the fungal hyphae. What causes the fungus to switch from this parasitic to a saprophytic stage and vice versa is not well understood. And it leads to sometimes idiosyncratic effects. So sometimes it works in the field, sometimes it doesn't. We have done a trial in a greenhouse and it didn't work. And once we started looking at its ecology, we found also through the work of another MSc student, Rui Carvalho, he started doing all these assays, and he found out that Peconia chlamydosporia is actually very susceptible to chemical pesticides. So they inhibit its radial growth and sporulation. So it's difficult to get it to work if it doesn't establish properly. Also, Peconia chlamydosporia only parasitizes eggs. And if you have nematodes moving along in soil, not in egg form, they can still infect the plant. So there is another way of trying to control these organisms through bottom-up control via the plant. And okay, in vegetable grafting is a new thing. Grafting has always been used for vineyards and orchards, but never for vegetables. And now it's been developed for tomato plants. So nematodes are in the soil, so they only attack the root. And if you have a resistant root, resistant rootstock, you can then unite it or graft it into your scion, your tomato plant that you want to produce. A number of rootstocks are available from all these different seed companies, and they are supposedly resistant to root knot nematodes, the mainly distributed ones, Meloido arenaria, incognita, javanica, but not to Meloido hapla, which is possibly because of the resistant genes that have gone into these rootstocks. Okay, so once you start looking at whether these rootstocks are protecting plants in the field, in greenhouse, we conducted a trial looking at different rootstocks, embajador, imperador, multifort, silex, and they didn't work very well. If you look at what's going on concerning plant-parasitic nematodes in the rhizosphere of all these rootstocks, you can tell that root knot nematodes, Meloido here in light blue, they're not really different from simple ungrafted plants. But there are lots of other different nematodes that we didn't expect. So we found actually this hemicyclophora is actually being suppressed by the rootstocks. Once you start looking again, because you have all this diversity of plant-parasitic nematodes, there could be some horizontal control. And we did find it between Meloido root knot, and hemicyclophora. They have an inverse relationship. More interestingly, if you look at the root knot nematodes themselves, they too have pasteria, these endospores, these spores attached to the cuticle. Now, pasteria is a very specific bacterium. It doesn't just attach to any nematode going by. It's specific at an infrapopulation level. We started looking at how many spores did all these root knot nematodes have. And we found differences in rootstocks nematodes coming from the rhizosphere, different rootstocks. So this means that in the rhizosphere of different rootstocks, the nematodes are different. These same root knot nematodes differ, and that's why pasteria is attaching differently to their cuticles. So this indicates there's some selective pressure there changing the phenotype of root knot nematodes. I know I don't have a lot of time, but I'll just skip through all these slides or just go through them very rapidly. Okay, this was in the fields that we did all the trials. So now we move to try to understand what's going on in this selective pressure. We move to controlled conditions, and we have the different rootstocks and the different species of root knot nematodes. And we can see from the damage that they cause to the roots that Meloido arenaria that was supposedly suppressed by these rootstocks is not at all. Hapla is being suppressed, and it shouldn't, because Hapla was not one of the target nematodes of these rootstocks. And Javanica is not being suppressed at all. So we find different differences to what these seed companies are telling you about what's going on, whether they can be resistant or not to nematodes. And once you look at the fecundity, the fitness of these nematodes that do go in, so not many nematodes go in and affect the plant, but those that do have lots of offspring. And we found out that actually within 1 or 2 generations, these nematodes overcome resistance of the plants. Once you start looking at how they interact, we started doing more finer experiments in which we've divided, and you can see here, a single tomato plant has its roots divided into 2 different pots. And in each different pot, we've added 1 species of nematode in blue, 1 in red. They're not actually blue or red, but we know what they are. And we're trying to find out whether they are competing either indirectly via the plants, so they're separated in different pots, but they're affecting the same plants, whether they're directly affecting each other's, or no, if there is some intraspecific competition as well. And what we find out is they are directly affecting each other's. So if you have them together in the same pot, you end up with less infection. This was a surprise to us because we weren't expecting these nematodes to attack each other's, but they're directly affecting each other's. So why is this? This is most possibly because, and this is the target, the objective of this project, the Handler project that we have now. We think they're communicating. They can tell each other's to go away or to come or to, you know, very recently, these ascaricides, these nematode pheromones have been discovered that affect nematode behavior in a number of ways. And these pheromones are produced by all sorts of different nematodes, that you can see here. So different, this is the only plant parasite here, but there's lots of different ones, parasites of animals and of insects and bacterial feeding, fungal feeding. They all produce these pheromones that have different behavioral effects, and they release them into the rhizosphere. And when they do release them, nematodes are not the only ones that can pick up these signals and react to them. Actually, different fungi and bacteria and plants can not only detect them, they can metabolize them. And you can see here ascaricide, that pheromone, ascaricide 18, that's being secreted by the nematodes and that attract nematodes. The plant is taking it up and metabolizing it into ascaricide 9. You can see it's just a matter of reducing the length of this fatty acid side chain here. So it's very simple transformation here. What it does is from 18 to 9, if you look at different proportions, ascaricide 18 attracts nematodes. And once you start having lots of ascaricide 9 that these plants are then releasing, you are repelling nematodes. So nematodes communicate. How this affects the rhizosphere and their interactions with all these other nematodes also and other organisms is an open field of research that we're very much interested in. So I've told you about nematodes, the good and the bad. I've exceeded my time, but I'm open for questions. And as for the ugly nematodes, I just want to tell you that beauty is in the eye of the beholder. They're all lovely to me. Obviously, thank you for listening. And of course, I didn't do all this work alone, and I had lots of help from different people in different points of the globe as well. And this was funded by all these projects listed here. So thank you for listening. Sorry for taking a bit long. Are there any questions?
Thank you very much, Sofia, for the fascinating presentation. So the time is really, yes, but I suppose that people can ask questions. We can have a few questions. I'm certain that all the information you gave us certainly will raise some.
Can I make a question?
Yes, please.
So by the last.
On your camera, please, Pedro.
No, I can't because the connection it's really bad if I turn it on.
Okay,
Sorry. So your last findings, it was that the plants have the capacity to synthesize that hormone. So we can say that that's the way we must to take to prevent these nematodes attacks.
It's a good question, and we are very interested in this. Ascaricides, this pheromone, is a very simple molecule biosynthesized by nematodes using basic metabolism, carbohydrate and lipid metabolism, which means this kind of metabolism is highly conserved throughout lots of different life forms. And plants certainly can use these ascaricides, transport them, change them, release them again. Plants detect the ascaricides, and only this detection itself or by itself can trigger what's called a pattern triggered immunity. So once it starts picking up ascaricides, plants know nematodes are there, and they elicit or they induce all these defenses that are generalistic defenses of using jasmonate and salicylate pathways, the plant defense pathways. But what has been found, and not by us, we're looking at this, but what has been found is that they not only do this, they actually take them up, change them into forms that repel nematodes. So they can communicate, they can tell nematodes to go away. Whether this then is then perceived by nematodes differently, and whether some nematodes are suppressed and others, or repelled and others aren't, you know, that's where the ecology comes in, and that's what we're trying to find out.
Thank you.
Any more questions?
Actually, I was telling you, not only plants can detect these ascaricides, we know that some fungi that are closely interacting with nematodes actually change their physiology as well, according to receiving these ascaricides. So we were thinking maybe even Poconia chlamydosporia, the fungus I told you about that switches from being a saprophyte to a parasite, might actually be responding to ascaricide presence as well. And this is something that we're very interested in, and we're going to look at certainly. Is there any further questions?
No, I suddenly I had all the screen black. Well, I don't know. Apparently, there are no more questions. I suppose I was told that the students have to be back in classes by 2 o'clock in the afternoon. So it's getting.
Yes.
A bit late. I thank you very, very much, Sofia, for this beautiful presentation. It is really a fascinating work. I am suspected, of course, but it's really a fascinating world. And I thank everybody for joining us in these seminars, and I see you again, hopefully in 2 weeks' time. Thank you.
Thank you, Celia. Again, thank you, everybody. Sorry for taking a bit long. Thanks for listening.